Hi there, welcome to this video. Um, in this video, we're going to have a look at what we call inhibitors of enzymes. Now, first of all, think about the language. What do we think inhibitors might mean? So if something is inhibited, it means we stop it from happening. So an enzyme inhibitor stops the enzyme from working. Now, sometimes it can be a permanent change. So, an inhibitor binds to the enzyme permanently, and that enzyme will not work at all. Sometimes it only binds to it temporarily, and then the enzyme can continue working afterwards. So, in that way, it's a difference between it being denatured and it being inhibited. It's simply the case that when an inhibitor is bound to an enzyme, it doesn't work. But if the inhibitor gets released, the enzyme will work again. When we looked at denaturing last time, once an enzyme's denatured, it actually doesn't work again. Okay, so it's more of a permanent thing. Um, so inhibitors are things that stop enzymes working. Now, the other thing to consider is that in a reaction, there isn't usually one enzyme. You've usually got lots and lots of enzymes working on the substrates to do the same thing. You know, we talk about enzyme concentration, don't we? Because it's not just one enzyme molecule. So, in the context of lots of molecules, if by adding an inhibitor into a solution, for example, you will reduce the activity of the enzyme. You probably won't stop it because it probably won't bind every single enzyme molecule. So it depends whether you're talking about a single molecule or whether you're talking sort of bigger picture, really. So, first of all, we're going to have a look at two different types of inhibitors. So, we've got what we call competitive inhibitors and non competitive inhibitors. Now, we know that the way an enzyme works is that the enzyme has what we call an active site. We need to make sure we're constantly using this language. And then we've got a substrate that has a that has a shape that's complementary shape to the active site. Okay, so we've got the active site of the enzyme, and we've got a substrate with a shape that's complementary to the active site. Now, if I add to that something called a competitive inhibitor, the competitive inhibitor would have a shape that's similar to the substrate. So you can see that this section of the competitive inhibitor is going to bind to the active site. So a competitive inhibitor has a shape that's similar to the substrate. It can't be the same as the substrate, otherwise it would be the substrate, but it's similar to the substrate and it's complementary to the enzyme's active site. So what will happen is that rather than the substrate binding to the active site, the inhibitor can sometimes bind to the active site. But this is my inhibitor binding to the active site. And you can see then that the substrate, oops, not a very good picture, the substrate can't, not much better, the substrate can't bind to the active site. They're competing for the active site. So when you've got a competitive inhibitor, the substrate and the inhibitor compete for the active site. Okay, because they bind in exactly the same place. So they compete to bind to the active site. Now, most competitive inhibitors are reversible. So that means that they bind to the active site and then they dissociate again. They come off again, and then the substrate can bind, and the inhibitor might bind again, the substrate might bind again. Remember, it's totally random. These molecules, inhibitors, substrates, enzymes, all have their own kinetic energy. They're moving around totally randomly, and it's random chance which one of them will bind to the active site. Adding more inhibitor will obviously decrease the rate of the reaction because more of the active sites will be blocked by inhibitors and the substrates won't be able to bind. Okay, if you increase the concentration of substrates, on the other hand, you will basically dilute the impact of the inhibitor. So increasing the concentration of the substrate, let me draw you it on a graph. Increasing the concentration of a substrate, put down here, substrate concentration, and then at the side, we put, one second, the rate of reaction. We always call it initial rate of reaction. It just means the rate of reaction at the start. If we didn't have an inhibitor, the initial rate of reaction, the rate of reaction would increase and then it would plateau. So that's with no inhibitor. Now, if we increase the substrate concentration, the rate of reaction will actually be, with a, I can't talk, with a fixed concentration of a competitive inhibitor. So let's do the next line in a different color. So with a competitive inhibitor, just with a fixed concentration of it, not varying it. But if we've got competitive inhibitor, we know that the rate of reaction is going to be less. We know the line is going to be underneath the pink line, don't we? Because we've got an inhibitor, it's going to slow down the rate of reaction. By having the substrate concentration then increasing, the higher the concentration of substrate, basically it increases the chance that the substrate will bind with the active site instead of the inhibitor binding the active site. So although the line is below the pink line, it's still going to be an increasing line, okay? Because the more substrate we add, the higher the chance that it will be a substrate that binds to the active site rather than an inhibitor. Without any inhibitor, it's always a substrate that binds, so the rate is much quicker. Okay, but with an inhibitor, the rate is slower because sometimes an inhibitor binds, but as we increase the rate, the concentration of substrate, we increase the chance that it's going to be a substrate that binds rather than the inhibitor, and therefore the rate is still increasing. In terms of the plateau, it will plateau at the same point. Okay, it will meet the other line, right? But it meets it much later. Okay, so we end up with a line like this, and you need to know the shape of that graph there. So increasing the substrate concentration effectively dilutes the effect of the inhibitor. If enough substrate is added, then eventually it's unlikely that an inhibitor will bind with the active site, and it will always be pretty much always be the substrate binding, and therefore the rate of reaction gets back up to where it was with no inhibitor at all. The second type of inhibitor that we've got is called a non-competitive inhibitor. So this one, as the name suggests, is not competing for the active site. Okay, it does not compete for the active site. So if this is our enzyme, and this is where our substrate binds in the active site, in this one we've actually got a slightly different setup. So our enzyme has another binding site somewhere else. That binding site can be anywhere on the molecule, anywhere on the enzyme, except the active site. And this is called an allosteric site. Okay, it means somewhere on the enzyme that it binds that is not the active site. So this site over here is still called the active site. But now we've got a second binding site called an allosteric site. And the non-competitive inhibitor will bind just here. Okay, so this is our non-competitive inhibitor. So it binds to the enzyme somewhere that's not the active site. And what that causes to happen is it actually causes the shape of the active site to change shape. And therefore, when the substrate then tries to bind to the active site, the substrate is no longer complementary and it can't bind. To the active site. Okay, so the non-competitive inhibitor binds to an allosteric site that alters the shape of the active site, and therefore the substrate is no longer complementary and cannot bind. Now, some of these non-competitive inhibitors are reversible, just like most of the competitive inhibitors. They bind and then they release. But some of them are irreversible or non-reversible, which means that once they bind to that allosteric site, they bind forever and that enzyme can't be used again. Okay, and that has a massive impact. If we look at the graph then and how it affects the rate of reaction, the graph looks very different. So if we recap what we did before, if this is our graph, remember looking at the initial rate of reaction, the rate of reaction right at the start. And we're looking at the impact of changing the concentration of the substrate. If we didn't have any inhibitor, our line looks like that. It increases and then it plateaus. We looked at what happens if we add a competitive inhibitor, and we saw that the line increases at a slower rate, but then it joins at the same plateau. So that was a competitive inhibitor. Oops, can't spell. So what then happens if we have a non-competitive inhibitor? So the more substrate you add, it's still going to cause this upward line, but it's going to be lower again than, well, it's not necessarily lower at the start. The very start might be the same. But what happens is that it's going to plateau sooner. That's the real significance of it. So it might go up with this one at the start, okay? But then basically it's with this one on the first time that an inhibitor binds to an enzyme, okay? Because that's effectively the same as a competitive inhibitor, because it's just stopped that enzyme working in the moment the first time it binds. But then what happens in the competitive inhibitor is because they're usually released, they can be used again. The non-competitive inhibitor, because they're often irreversible, it means that they can't be used again, and therefore the rate can never get up here. It actually plateaus much sooner. So the maximum rate is controlled by the number of inhibitor molecules present. However many substrate molecules you add, it's still going to plateau at a much lower rate. So you need to learn the shape of those different graphs. Okay. Now, what we've looked at then is two different things. We've looked at competitive, which bind to the active site, versus non-competitive, which bind to the allosteric site. And we've also looked at how we've got reversible inhibitors versus non-reversible. inhibitors. Okay, so make sure you just know the difference between those. Um, the third thing that we're going to look at in this session is something called an end product inhibitor. So an end product inhibitor is one that works in a series of reactions. So let's say we start with this substrate, I'll just call it S, and a reaction happens that's catalyzed by enzyme number 1. So we'll call that enzyme 1, and it produces a product. Now, that product might then be the start of another reaction. So, although it's the product of a reaction catalyzed by enzyme 2, it might then act as the substrate of a reaction catalyzed by enzyme number 2. So, in another enzyme, the product of this reaction, even though it's got a different shape to this substrate, the shape is now complementary to the active site of enzyme number 2. So it can act as the substrate for another reaction. And we can get a whole series of reactions happening like that, where the product of the first reaction is the substrate of the next, and the product of that is the substrate of the next, and the product of that is the substrate of the next. And then we get our final product. So we've got this series of enzyme catalyzed reactions, and that's mostly how reactions happen in our bodies. We don't often just have one reaction. We often have a series of smaller reactions where something happens in a number of stages. Now, each of these enzymes is going to have a different shaped active site. I've not really drawn my enzymes so they fit in, but they're each going to have an active site that's a different shape, aren't they? Okay, because that's how enzymes work. So then what's going to happen is this final product, if we give these numbers, for example, if we said this is number 1, or we call it letters, our enzymes have got numbers, so let's call them A, B, C, D, E, F. Molecule F here might then bind on an allosteric site, or it could be a competitive inhibitor, in fact, it could be either, but it acts as an inhibitor to inhibit enzyme 1. So if the last product in the series of reactions went back to bind to the enzyme that catalyzed the first reaction, that might then change the shape of the active site of enzyme 1, and therefore enzyme 1 can't catalyze this first stage of the reaction. And therefore, it stops this reaction going on forever. We don't want a huge build-up of the same molecule. We need to have some control of the molecules that we're making. And therefore, by the end product acting as an inhibitor for an earlier stage of the reaction, we can actually control it and make sure we don't get a huge build-up of some kind of molecules. Okay, so we call that end product inhibition. Now, the last thing that you might read about on here is various examples. Now, the specification doesn't ask for specific examples, but they are going to use examples in the question, so it might be worth you having looked at some of the examples. So I have a little outline of a few now, but they are not something that's listed in the specification for you to be able to recall. Okay, so one very famous example is a chemical called cyanide. Now, cyanide is a non-reversible, so they're not going to ask you to recall that cyanide is non-reversible, okay? It's just have an awareness of them. So it's a non-reversible inhibitor of one of the enzymes used in respiration. Now, we know how important respiration is, don't we? Because respiration is how we release the energy from glucose. Okay, so it's a non-reversible inhibitor of an enzyme in respiration. So if it stops respiration from happening, then we can't release ATP. or we can't release, we should say, you should always say release energy and make ATP. So it can't release energy or make ATP from the glucose. And ultimately that can kill cells. If cells don't respire, they die. Okay, so it will kill cells and it could actually be fatal for the person. It is actually poisonous. Okay, so cyanide is a really, really famous example of an inhibitor. Another really famous one is aspirin. So I'm sure you've heard of tablets, aspirin, like paracetamol, aspirin. You'll have heard of it. It comes in a similar box. Okay, so aspirin is also an inhibitor, and it works as an inhibitor to reduce the pain that we're experiencing. Okay, another really famous one is penicillin. So penicillin is an antibiotic, and that works with an inhibitor. Another one would be, sorry, I'm trying to find all the lists just to see which ones come up most often. Another one is an ATPase inhibitor. Okay, so ATPase is making ATP. So there's lots of examples, and you can research a little bit about each of those if you want to, but you don't have to recall them. But just be aware that in the exam, you need to know for definite competitive, non-competitive, how they work. You need to definitely know the shapes of the graphs. You need to know the difference between reversible and non-reversible, and they're likely to incorporate that into examples. So it might be worth a little look at how some of them work, but don't worry too much about memorizing it all. Okay, and that's the end.